So this is going to be why we can't go without oxygen, right? We can't go without oxygen because we have to be able to make ATP. So that's just the way that we work. All right, so let me widen up the screen here. Actually, I should probably get my pen going first. Okay. All right, so what we have here is we've got 3 stages of aerobic cellular respiration. Glycolysis is the first stage. Okay, the Krebs cycle is the second stage, and then the electron transport chain is the third stage. Now, it's not like one glucose molecule is in one mitochondrion and like it has to happen very slowly, one step at a time. Like this is all happening simultaneously, but with one glucose molecule, that one glucose molecule starts at glycolysis, then goes to the Krebs cycle, then goes to the electron transport chain. Simultaneously, other glucose molecules are at different parts of the stage. Okay, because it's more than one glucose molecule is being processed at once. So, what does glyco make you think of? Okay, so it should make you think of glycogen and glucose. So, glyco should make you think of glucose, and then lysis is to break or to cut. Okay, so glycolysis is where we're going to cut glucose in half, basically. Okay, so what I did was here, I took a glucose molecule. Whoa. I took a glucose molecule and I basically like straightened it out. You know how glucose actually has, it's like 6 sides and it's like, it's a hexagon and then it's got the carbons and the hydrogens and the oxygens coming off. Well, what I did was I just took like the ring there and I like broke it and stretched it out in a chain to make it easier. Okay, so that's what I did there. And I can draw a picture for you guys of that for you guys in a little bit. So what's going to happen here is that we need to break this glucose down. We've got to get the energy from the bonds. So what's going to happen is that hydrogens, so there are hydrogens and oxygens like all attached here, right? I just didn't draw them all on here because that would be really complicated. But you guys know that in a glucose molecule, there's a bunch of hydrogens and oxygens attached. Specifically, there's 12 hydrogens and 6 oxygens. So one of the hydrogens that's over here and one of the oxygens that's, and one of the hydrogens that's over here are going to break off. So we have 2 hydrogens that are going to be broken off, and they are going to attach to a carrier molecule. Y'all remember from photosynthesis how NADP was a carrier molecule? Well, the main carrier molecule in glycolysis is called NAD. No P. All right? NAD+. All right? And what's going to happen is that hydrogen and its oxygen, that hydrogen and its oxygen, that hydrogen and its electron are going to bond to NAD to make NADH. Okay, so we had 2 NADs come in. They each grab an H, and now we have 2 NADH. All right, those 2 NADHs are going to go to the electron transport chain. Okay, now I need to kind of backtrack a little bit because I should have explained this at the beginning. But glycolysis happens here in the cytoplasm, and the Krebs cycle happens here in the mitochondrial matrix. And then the electron transport chain happens in the cristae. Okay, so that's my bad. I didn't talk you through the parts. So we've got, here's the mitochondrion, right? Okay, and we've got the cristae, which is the membrane folds. And we've got the outer membrane, and then we've got the matrix, which is just the space inside.
So does it happen in the cristae or the matrix?
So let me talk you through it. So the glycolysis happens in the cytoplasm, outside the mitochondrion. Okay, the Krebs cycle happens in the mitochondrial matrix, which is this fluid area inside of the cristae. And then the electron transport chain happens in the cristae. Cool? All right, so we got cytoplasm, we got the outer membrane, which is a phospholipid bilayer. We've got the cristae, which is another phospholipid bilayer, but it's all convoluted. And then there's the matrix, which is inside the cristae. Everybody's cool with that? Sorry, I should have outlined that to begin with. So glycolysis is happening. We have a glucose molecule. Glyco means glucose. Lysis means to break. So we're breaking this glucose in half. In order to do that, two NAD molecules, which are carrier molecules. They're going to come in and they're going to pick up a hydrogen from two different parts of the glucose molecule. So two NADs come in, pick up two hydrogens and their electrons, and take them to the electron transport chain. Where do you see the electron transport chain? In the cristae. Okay, so those NADHs are going to the cristae. They travel. Okay, what's going to happen next is that ATP is coming into play. We're going to have two ATP molecules that are each going to drop off a phosphate and then leave as ADP. So we have ATP come in here, drops off a phosphate, leaves as ADP. Same thing happens here. So we used two ATPs there. Okay, the reason why we're doing that, remember that breaking that phosphate off is you're releasing energy there. So we're basically energizing the next part of this reaction. That's why the phosphates bond. Okay, so those phosphates bond. The binding of those phosphates is what causes the actual lysis of the glucose molecule. So you see right here, the bonding of those phosphates causes the glucose molecule to break straight down the middle. So now instead of having one 6-carbon molecule of glucose, we've got two 3-carbon molecules. Each of those is called a pyruvate, also known as pyruvic acid. Pyruvate is also known as pyruvic acid. Mark, this is not writing very well today. Okay, so now that the phosphates have done their job and have broken the molecule in half, what do you think is going to happen to them?
So wait, what is a pyruvate?
These two 3-carbon molecules that are left over when the glucose breaks down the middle, those are each called a pyruvate.
Like with the phosphate too?
Uh-huh. Yeah, but we have to break off the phosphate, right? We can't just leave the phosphate there. The phosphate's not helping anymore. It already did its job. We want to be able to use the phosphate somewhere else. So what's going to happen is, what's going to come pick up the phosphate? ADP, right? ADP molecules are going to come back, pick up their phosphates, and leave as ATP. All right, so the ADPs come back and leave as ATP. So we used two ATPs right here, but then we got them back here. So now we're at like 0. Like we used two ATPs, we made two ATPs, so we're at net of 0. And we have two pyruvate molecules. Those two pyruvate molecules are going to travel into the mitochondrion, okay? On their way, okay, two ADP molecules are going to come in and pick up two more phosphates. There's another somewhat complicated process that happens here called the link reaction that we don't have to learn about. So just know that two more ATPs are made here as the pyruvates are traveling into the mitochondrion. Okay, so we used two ATPs right here. So this was -2. This was +2. We got them back right here. And then we got +2 here as well. So how many net ATPs were made during glycolysis? Good. We had two net ATPs made during glycolysis.
So where does the second 2 come from?
This right here?
So,
Is that what you're asking?
yeah. So,
We're not learning about the steps of this. Just know that two more ATPs are made. Okay? All right, so now we have two pyruvate molecules that are moving into the mitochondrion, okay? As part of the link reaction, that we're going to break off a carbon off of each of those pyruvates right here. A carbon is broken off of each of those pyruvates. What's going to happen is that oxygen is going to come in, pick up one of those carbons from one of the pyruvates and leave as CO2. Happens to the other one as well. Oxygen comes in, picks up this carbon from this pyruvate, and leaves as CO2 as a byproduct that we exhale. Okay, so now we have two carbon molecules called acetyl-CoA. Acetyl-CoA is going to start off the Krebs cycle. the acetyl-CoA molecules, we have 2 of them, and they're going to go in one at a time. Just like how we have to do 2 rounds of the Calvin cycle in photosynthesis, we have to do 2 rounds of the Krebs cycle. Does anyone remember another name for the Krebs cycle? I'm sorry. Remember me calling it the citric acid cycle too when you were a freshman, maybe?
I don't.
Okay, so the Krebs cycle is also known as the citric acid cycle. So maybe make a note of that. Citric acid cycle. Okay, so the Krebs cycle is going to start off with one of the acetyl-CoA molecules. That acetyl-CoA molecule is going to bond in the mitochondrial matrix to a 4-carbon molecule that's waiting there. This 4-carbon molecule, we already know. This 4-carbon molecule is called oxaloacetate. And I'm not going to be able to write that on here because it's way too... Oxaloacid. Mm-hmm. Yep. Mm-hmm. Okay, so this acetyl-CoA bonds to oxaloacetate, which is a 4-carbon molecule. What's 2 plus 4? 6. So we made a 6-carbon molecule, which is called citrate. This 6-carbon molecule that's made when an acetyl-CoA bonds to an oxaloacetate in the mitochondrial matrix is called citrate. That's why we call it the citric acid cycle. Okay, it's also known as citric acid. Citrate, citric acid. Kind of like how it was malate and malic acid. So citrate is then going to have one of its carbons taken away. Okay, oxygen comes in, takes away one of its carbons. Okay, every time that a carbon is taken away, a hydrogen is also taken away that's attached there at that carbon. So this NAD is going to come and pick up a hydrogen. So at this step, an O2 molecule comes and picks up a carbon, and an NAD molecule comes and picks up a hydrogen. Where do you think the CO2 goes? It's exhaled. Where do you think the NADH goes? Where did it go before? It went to the electron transport chain from glycolysis, right? Okay, so the NADH is going to travel over to the cristae for the electron transport chain. Okay, now we have a 5-carbon molecule. Okay, but we're not going to stop there. This next carbon is going to be broken off. So an oxygen comes, picks it up. Now it's CO2. That's going to be exhaled. NAD comes and picks up the hydrogen that also breaks off, and then that NADH is going to go to the transport chain. Okay. All right, so now we have a 4-carbon molecule with some hydrogens and oxygens attached. I did not draw all the hydrogens and oxygens that are attached on any of these molecules because it would be so crazy confusing to look at. Okay, so what you're looking at here is 4 carbons with a hydrogen attached to each, and I just kind of expressed that so that you would realize that there are hydrogens and oxygens attached here still. Okay, so when that second carbon is taken off, 1 ATP molecule is made because an ADP comes and picks up a phosphate. Okay, so you got 1 ATP molecule made. So we made 1 ATP, we made 2 CO2s, we made 2 NADHs, and now we have a 4-carbon molecule left. Okay? What's going to happen now is that carrier molecules called NAD and FAD are going to come and pick up the remainder of the hydrogen molecules. Where do you think they're going to take them? To the electron transport chain. Okay? So what are we left with at the end? Now that we've removed 2 of the carbons, we've removed a bunch of the hydrogens, what are we left with? A 4-carbon molecule called oxaloacetate, so that we can start the process again, right? Okay, so now this second acetyl-CoA is going to come in and start the cycle again. So this is going to happen 2 times per glucose molecule because we have 2 acetyl-CoA per glucose molecule. So how many ATP are going to be made during the Krebs cycle for 1 glucose molecule overall? 2, right? Because each round, for each acetyl-CoA, we make 1 ATP molecule. So technically, we make 2 overall. Okay, so far we've got +2 from the glycolysis and +2 from the Krebs cycle. Okay, now the next step, and the step that makes by far the most ATP, is the citric acid cycle. I'm sorry, is the electron transport chain, which is happening in the cristae, which is this convoluted membrane over here. So I kind of drew them as little dots because they're so small, but these are proteins embedded in the membrane here. Okay, kind of reminds you of the proteins embedded in the thylakoid membrane, except for this time they're embedded in the cristae of the mitochondrion. So this is going to occur similarly to the electron transport chain that was part of photosynthesis, except for this time what's happening is that hydrogens are moving through these proteins that are in the membrane, and as they move through, their electrons are breaking off and are moving through the proteins. Okay, so hydrogens are moving out and into the space between the outer membrane and the cristae. And as that is happening, their electrons are breaking off and are traveling from one protein to the next.
So, do the hydrogens leave?
They're not leaving, they're hanging out in this space right here because they're going to come back in. Okay? So the hydrogens are moving out through the proteins. As they move through the protein, their electrons are coming off. So now we have hydrogen ions out here, which are essentially just protons. Okay? And then electrons that are now moving from protein to protein with the gradient. Okay? Once the hydrogens build up here in the space between the cristae and the outer membrane, when we get to this last protein, this is ATP synthase again. We also have ATP synthase enzymes in the mitochondrion, just like we do in the thylakoid of the chloroplast. Okay? So this last protein here, and we'll get this zoomed up later on, but right now we're just kind of talking general. Okay? We've got this last one is ATP synthase, and the hydrogens are going to do what do you think they're going to do with the ATP synthase? Move through it. They're going to move through it back into the mitochondrial matrix. Okay, but this time, different from photosynthesis, they're going to bond to the oxygen molecules that are waiting out here. When we bind two hydrogens to an oxygen, what do we get? Water, okay? So we're making water here, and then what do we know happens to phosphates and ADPs when hydrogens are rushing through and there's a concentration gradient change? What do? They bond to each other, right? So as these hydrogens are rushing back into the matrix through the ATP synthase, that's going to cause two things to happen. It's going to cause the hydrogens that are rushing in to bond to the oxygens. It's also going to cause the ADP and phosphates that are on the outside to bond to each other. So what are we making here? Water and ATP. And we're going to make anywhere from 32 to 34 ATP. So plus 32 to 34, just depending on the movement. Okay, so I'm going to stop the video here.